Welcome to the front row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps. I'm alone in my office. And thank you all for joining us on this webinar to discuss the coronavirus as seen through the eyes of Kristian Andersen. So the front row, as you know, is a lecture series at Scripps. That's an opportunity to update the community on highlights of what's going on. And I think it's particularly timely right now. We'd like to thank at the outset that we have KPBS as a sponsor for this event. And also, I'd like to thank many of the regular attendees of the front row who are our donors. This is not a fundraising event. We're really primarily here to provide information about coronavirus to the community. But we do want to thank all of you who have in fact supported our mission. So thanks very much. And really, I want to focus on the coronavirus, and really the highlight is Kristian Andersen. But I think there's a lot of new people that are new to Scripps that might be joining the webinar. Our auditorium, the largest auditorium on campus, holds 300 people. We are at 1,300 attendees to this webinar. So this is really a remarkable response. This is unprecedented, and it's unprecedented times indeed. So I'm reflecting a little bit on the idea of this webinar. I mean, people enjoy social gatherings. We like to come and hear a lecture. We socialize with each other at the beginning and at the end. We share an experience in the audience. We laugh together. We share wonder together. And the speaker will interact with the audience and react in real time to what's going on. And this webinar format presents some real challenges. And I think our new reality is in fact going to be a virtual reality for quite a while. And I think perhaps the most awkward thing about this is it's customary at the end of the lecture for everyone to clap. I don't know what to do about that. I think it's okay to clap all by yourself. So just go ahead and do it at the end. So there will be a Q&A at the end, and the Zoom application has a Q&A button, and you can type your questions in there, and we have a moderator that will do the best they can to try to field questions for Kristian at the end of his presentation. So our speaker today, sorry. There we go. I thought I'd start out by giving a little bit of information for the new people at the Scripps Research Institute. So Scripps has been around for almost 100 years, and we have two campuses. One is here in La Jolla, and we have another campus in Jupiter, Florida. There are 200 faculty members at Scripps on both campuses working on basic and translational research. And you're just going to hear a sampling of that today. One of the things we're most proud of is that we have a nationally ranked graduate school. So we're ranked consistently in the top 10 by US News and World Report in both chemistry and biology. And one of the things I want to convince you of is that Scripps is a long history of working on critical viral problems. And much of our ability to pivot and rapidly respond to the coronavirus outbreak is based on decades of experience that the faculty here have on working on such viral pathogens as HIV, flu, and many other pathogens. So many of the techniques that have been applied to this put us in a great spot to attack the coronavirus. There have been a number of drugs on the market now that have been discovered or developed at Scripps or developed using Scripps technology. Most recently, in March, the drug ozanimide that was developed at Scripps was approved by the FDA for treating multiple sclerosis. So we're very proud of this and many of the other accomplishments that you can see on this list of medications that came out of Scripps. It's one of our missions to coordinate the global health. So Kristian Andersen in particular has coordinated all of the faculty working on infectious disease at Scripps into what we're calling the Global Health Initiative. And this is a collaboration between three arms of Scripps, the Research Institute, the Caliber Translational Research Institute, and then the Scripps Translational Research Institute. Caliber is led by our president, Pete Schultz, and SRTI is led by Executive Vice President Eric Topol. The three pillars of the Global Health Initiative are surveillance and diagnosis, production of vaccines, and antiviral therapeutics. Now, diagnostics are well in hand. Basically, you can do PCR diagnostics for viral RNA. We can detect antibodies in our blood from infections. That's all well worked out. The challenges there are primarily on scaling up to test 300 million people. But Kristian's expertise is in surveillance and analysis of viral sequencing and tracking. We have tremendous expertise in vaccine and then developing antiviral therapeutics. Kristian has gotten a lot of media attention in the past weeks. And I'm sorry, my slide's automatically advancing, just bear with me. Kristian published a seminal paper on the origins of the coronavirus, and it has gotten all kinds of media attention. I won't say more as he'll give you the full story there. Eric Topol's laboratory is also working on ways to tell if you are infected, and it turns out you can harness personal fitness devices, and the biometric data they collect can be used to inform you that you might be experiencing some kind of viral event. So this could really be a tremendous way to crowdsource human health, and Eric is at the forefront of digital medicine. We have lots of work going on for antivirals, and here's some vaccine work. So Dennis Burton is now screening for antibodies from infected patients and looking for those antibodies that might serve as neutralizing antibodies that could be used to treat infections or possibly in vaccines. Ian Wilson has been using X-ray crystallography. They determined the structure of an antibody in combination with that was isolated from a patient, bound to the spike protein on the surface of the coronavirus. And that's giving some clues as to how you might target this virus with drugs. Andrew Ward's lab is looking at the structure of the glycoproteins on the surface of the virus in combination with antibodies. And the Farzan and Cho labs at the Florida campus are actually working on finding compounds that inhibit viral replication. And I want to point out that Mike Farzan will be giving the front row from Florida next week. So if you're interested to hear more, you can tune in next week. And finally, on the antiviral therapeutics, I want to highlight there are many things going on, but I want to highlight the work going on at Caliber. So Arnab Chatterjee is leading the charge on application of screening to the Reframe library. This is a collection that they built of every single compound that has ever been introduced in humans as a drug or in clinical trials. And what they can do is rapidly screen through these libraries and look for compounds that are antivirals, that inhibit viral replication or viral entry. And in addition, they're screening for compounds that could be used in combination therapies, such as those that were particularly effective for HIV. And the value of this is all of these compounds have been in human beings, and if they're proven effective in combating coronavirus in the lab, they can be rapidly deployed into people because they've already been shown to be safe. So I hope I've convinced you that there's a lot going on at Scripps. We have a global health initiative approach to combating viral diseases. And with that, I would like to turn it over to our featured speaker, Kristian Andersen, and he's going to be telling you how he has been approaching tracking the virus and then learning how we can outsmart the virus in this time of global pandemic. So thank you, enjoy the webinar. We'll have questions at the end, and I'm going to turn it over to Kristian for the talk.
Thank you very much, Jamie. And thank you everybody for tuning in. I should say before I get started here that like anybody working in global health and infectious diseases, we knew we were going to have a pandemic at some point. But I have to admit that I did not expect that that would result in me having to give a talk for 1,500 people from my living room. But here we are, and welcome to my home. So as Jamie mentioned, we've been looking at very many viruses over the years, coronavirus being one of the latest ones. And just to introduce the type of work that we do, I just want to start with a little animation here, which is going to take a little while to run. So what I'm showing you here is the spread of SARS-CoV-2 out of China and then across the world. This is based on genomics. So just by looking at sequences of viruses, it also means that it's undersampled. So there's some countries you might not see lighting up yet, but they will as this pandemic get going. So the way that we do this is that we do that via a main framework, which is called Genomic Epidemiology. So the way that this works is that you can imagine that a patient comes into a ward somewhere in the world. It could be here in San Diego. It could be some other country. A blood sample or a sample, in this case, a sputum sample or nasal swab is taken from that patient. And then that sample is taken to the lab where the virus is sequenced. So everything that's in that sample gets sequenced on sequences, and from this we get the virus specifically infecting that individual. Now, viruses, especially RNA viruses, they have little mutations in the genome as they spread between us. So this means that from every patient we get this, the viral genome is now a little blueprint of exactly what did the virus look like in that patient. We can take that information, we could piece it together if we have time information and we know where the virus is actually collected. We can take all of that information and create the kinds of animations understanding the spread of the virus as I just showed you for SARS-CoV-2. So these are the kinds of questions that we're interested in. We're interested in using genomic epidemiology to understand how do these viruses spread? Where do they come from? How do they evolve? And importantly also, what are the kinds of factors that are actually leading to spread? So what factors might try and prevent the spread of these viruses? Which factors might actually promote the spread of these viruses? As Jamie mentioned, we have been working on quite a number of viruses over the years. I sort of got started in this type of work a little over a decade ago, focusing on Lassa. That work got expanded to Ebola. More recently, looking at Zika. We have some projects on West Nile too. And then more recently, looking on SARS-CoV-2. So we have a lot of different viruses that we have been looking at, trying to answer these questions of how do they spread, what are the factors leading to spread. And I should say that a lot of this is hugely collaborative projects. So you'll see a couple of different icons down here. This is just for some of the projects that we are part of. But generally speaking, these are highly collaborative projects, typically with scientists from the countries that are actually affected from these viruses. So I'm going to show you on our work on SARS-CoV-2, but before I do that, I just wanted to mention that I'm giving a talk here from my living room, but actually my lab are the ones doing the real work here. I'm not actually doing anything. I just have a keyboard and a mouse in front of me. And they're actually the ones doing the work on a day-to-day basis. Many of them in the lab right now working on COVID-19 projects, many of them also working from home, working on COVID-19 projects, as well as all the other projects that we have going on Lassa, in Ebola, and all the other viruses. So just wanted to make it clear that everything that I'm presenting today is really their work. It is not my work at all. So as part of this ongoing pandemic, the San Diego community specifically has also really come together. So these are some of the new projects that we are part of, collaborations here with UCSD, with collaborators at Scripps Research at Rady Children. These projects are more focused on understanding SARS-CoV-2, COVID-19 here in San Diego, and with an initial focus on understanding healthcare workers. We know that healthcare workers often get infected with this virus, and we want to understand exactly why is that, what can we actually do about it. So these are some of the new projects that you're probably going to be hearing about in the near future. So let's talk about this. So this is a little screenshot that I took earlier this morning. It's of course the number of cases from the Johns Hopkins dashboard showing the number of total confirmed cases of COVID-19. We just rounded 2 million confirmed cases this morning. But I should say that by this time, the counting is way, way off because there's only so many cases we can actually diagnose. So the true number of cases is going to be a lot bigger than what we are seeing here with the 2 million people. And of course, as we all know, this is over the last just a couple of months here, this virus has very rapidly spread across the world. So just a few things about, this pandemic itself. So I mentioned these terms a couple of times. Most of you already know this, but the disease itself is called COVID-19. The virus causing the disease is called SARS coronavirus 2. And the virus was first detected here in the city of Wuhan, which is in China, Hubei province, as you can see here. Detected at the very end of last year, so at the end of December, and then immediately recognized as a rapidly emerging outbreak in the beginning of this year. Interestingly, if you look at where SARS itself, so it's not SARS-CoV-2, but SARS itself, was down here in Guangdong just about a decade ago or so, also came out of China. And it's a very similar virus to this one. So the virus is a beta coronavirus. You've probably heard this many times. It has an RNA genome, which means that we can count these little mutations. It's a pretty big genome. It's 30 kb. It's typically bigger than most other RNA viruses. The detection itself was this Wuhan market. And I'm going to talk a little bit about this because this is why we detected it, but we're actually not totally sure that this is where it got started. There are not of the virus, it's just the average of number of cases that will come from one case that's infected is in the order of 2 to 3. And just to give you a little bit of a comparison here, this is actually exceptionally high. If you look at flu, for example, it's only about 1.5. And what's really important about this number is that as long as this number stays above 1, now becomes the RT, the effective reproductive number. Once that starts above 1, that means that the pandemic will spread. What we need to focus on is to get that below 1, because once we can start getting this below 1, that means on average, 1 case is going to infect less than 1 case. The pandemic itself will actually start to contract. The case fatality rate of this is pretty high. It's much, much higher than seasonal flu, for example. We don't really have good estimates for this now. It's probably about 2 to 3%. The infection fatality rate, which is the number of people, all the people that get infected actually die from disease, seem to be about 1%. And massive bias towards elderly population as well as people with comorbidities. So that's obviously a big problem, but one really big problem with this virus too is that the number of severe cases, the people that get infected leading to severe cases, is very high. It's about 15% or so of those requiring hospitalization. So that's a very, very high number. And this is one of the reasons why we have seen, hospitals overrunning in Europe, in New York City, many other places in the world. So when we just look at this virus itself, some of the spread that we have via this is via droplets. So this is the primary way that the virus is spreading. Somebody sneezes on you, or via surfaces, somebody has sneezed on a surface and then the virus is stable here. And this is really the important bit why we've been talking about social and physical distancing. This is why this has been necessary, because the virus spreads so easily, we need to get away from each other. If we're close together, then chances are we'll get infected. That's the reason why we have to do social distancing. The reason why it can spread via surfaces is why you're told you have to wash your hands and don't touch your face. That's again because whatever surface you're touching might actually have the virus on there. And now one thing that's very special about this is very unfortunate is that we have a very significant amount of asymptomatic transmission. This is people that both don't actually get disease but are infected, but also people that are during the incubation period, so don't have symptoms yet. That's the time when the virus is really spreading. And this makes it very, very hard to sort of prevent the spread of the virus, because it means that somebody who is not actually sick yet is able to spread the virus to others. So that's a huge problem just in terms of stopping the spread itself. Now, the common symptoms here, things like dry cough, loss of smell, fever at some point, certainly the breath, things that we pretty often see. So these kinds of symptoms, especially the dry cough and the loss of smell, is not something we typically see in other diseases. So this seems to be pretty unique to this particular virus. Now, one thing I want to make clear is that because of the transmission of this virus, if we just let it go in our society, what's going to happen is that we're going to get many, many, many more cases than our healthcare system can actually handle. That means a lot of unnecessary deaths. It means that excess deaths will happen because people can't get to hospital, but also because they can't actually get treatment. So this is the whole idea behind this flattening the curve, as you have heard in many places. We need to flatten this curve. We need to try and prevent new infections from happening. And specifically, the way that we've been doing this is via social and physical distancing, wearing face masks, wash your hands, and so on and so forth. Okay, so with all of that out of the way, let me go into some of the work that we have actually been looking at. And I would like to split this into a couple of different things. I want to like to talk about what was before this, what is here now, and what we actually think we're going to observe in the future. So when I'm talking about before, this was before we was basically interested in a single question. How did this start? How did this actually all happen? And as I mentioned already, the way that this was detected was at this Wuhan market in Wuhan in Hubei province in China. The timing was that this was detected in December, about mid-December or so is when they realized that there were some cases linked to this market. I should say that this is during influenza season in China. So the fact that they can manage to detect a novel pathogen, something never seen before, figuring out that it was a coronavirus, let alone sequencing the virus and figuring out it's an entirely new coronavirus in the matter of just a couple of weeks, is totally unprecedented. This is very, very, very fast. Typically, these things take months. This time, it only took a couple of weeks. Now, we know that this was detected in December, but one of the questions we were interested in to understand is that, yes, but is December actually when this got started? Or is it possible that it got started earlier than that? So the way that we can do this is that we can look at the genomes of the virus. We can get an evolutionary rate of the virus, essentially a molecular clock, which tells us how fast is this virus changing. And this virus is like many other RNA viruses. It has about a 10 to the minus 3 evolutionary rate of substitutions per site per year. Very, very similar to what we're actually seeing in other RNA viruses. Once we have that information, we can estimate, okay, so when did it actually get started? And what we find is that this got started right around the time where it was also detected. Mid-November to mid-December or so is when we actually think this outbreak got started in the first place, which I said is very, very consistent with what we know from epidemiological findings and also from the initial detection. So the timing is not something that's been going on for a long time, very recently. But that led us to the next question, which was, well, where is this coming from? And again, we can do this via the genomic sequences. We are using publicly available genomes. All these earlier ones were from Chinese researchers. But what we can see is that, okay, where does this actually fall together with other viruses like it? And what's pretty clear is that there's lots of other viruses, just like SARS-CoV-2 here, that circulate in bats. They're not actually exactly the same virus, they're slightly different, but they're similar type of viruses. So because of that, we know that the ultimate reservoir of this virus, SARS-CoV-2, is bats. What we don't know is that, well, is there an intermediate host? So how did it actually get into the human population? I'm going to show you a couple of speculations on that. There might be an intermediate host. Pangolins, for example, have been suspected, but we don't actually know if that's the case. So reservoir was bats, so this came from some sort of animal. Led us to the next question, which is, but how many times did it actually get into humans? Was this a single event, or was this something that happened multiple times? And what we can show quite clearly is that this is a single thing. It's just happened once, the virus got into the human population, and then from there took off via human-to-human transmission. In the beginning of the outbreak, this was a little mysterious because we thought human-to-human transmission was limited, but the viral genomes, because we see so little diversity in the early viral genomes, really tells us this was a single event followed by human-to-human transmission. And indeed, when I'm talking about human-to-human transmission and spread, we can actually show this exactly just based on the genetic data, exactly how this viral population has been expanding over time, very similar to this exponential growth that we have seen in human cases. So we managed to answer these questions very early on, and so did many other researchers, and figuring out where did it come from, when did this all start, and how is it actually spreading. But the next thing we were interested in was looking at the genome, looking at the virus itself, and figuring out why is it that this virus seemed to be so unusual. Zoonotic viruses, except sometimes some flu viruses, zoonotic viruses don't just jump from an animal reservoir and then take off in the human population. These viruses don't know humans, so they're not really used to spreading amongst humans. So why was it that SARS-CoV-2 was so unusual in its ability to come as a zoonotic viruses and then immediately take off? So just to look at that, the way that we can see this is that SARS-CoV-2 here is that it can spread really easily. I showed you the R0 2 to 3, that's very, very high, and it can also cause severe disease. If we compare that to the flu viruses, for example, we see seasonal flu, which typically causes pretty mild disease. It's also infecting your upper airways. Upper airways, that's why you can spread really easily. If you look at things like bird flu, and when people rarely get infected by them, they lead to much more severe disease, but now there's infecting much further down in the airways and into the lungs. That's why you get the severe disease, but then the upside is that then it doesn't spread very well because it's not higher up in the airways. When we're looking at SARS-CoV-2, it can do both. It's capable of infecting the upper airways, so it can transmit really easily, and it's also able to infect lower down into the lungs, meaning that it's capable of causing very severe disease. So that was a mystery to us, and we just asked a very simple question, and that is, why is that? How come that SARS-CoV-2 can do that when we haven't actually seen that happening before? And the way that we addressed that was to say, okay, let's look at the genome. Let's look at the genome of this virus, and let's actually look at how this is different than other viruses, other SARS-like viruses. So what you're looking at here is just a genome of this particular virus here. What you can see is the single gene here, which is called the spike protein. The spike protein is the protein that binds onto the receptor on human cells. So this is what allows it to actually infect cells and cause an infection of the individual. So this is all mediated by the spike protein. If we look inside the spike protein has a very specific region here, which is called the receptor binding domain, or the RBD. This receptor binding domain is what specifically binds to the human receptor. It's called ACE2 is the receptor. This is the part of the viral spike protein which mediates that particular interaction. And what we find is for this particular virus, well, it has two things which we haven't really seen in these viruses before. First of all, it has the RBD, the receptor binding domain down here, seems to be highly capable of binding to the human receptor, and specifically to the human receptor. Doesn't seem to be very good at binding to the bat receptor, for example, which I think is the reservoir, right? So that was somewhat unusual, saying, why is that? The other thing that we found is that it has other part of its spike protein. Here it has an insertion of a little site here, which create what is called a polybasic cleavage site. These viruses, when they infect cells, they infect and they bind to cells via this S1 subunit. Then they get internalized into the cell, and now they need to cleave this protein in the middle here. So the S2 subunits can actually allow the virus to fuse into the cell and infect the cell. And what's interesting about SARS-CoV-2 is it has a specific site that mediates this. Things like SARS, for example, is actually not very good at that, but this one has this little site that makes it highly efficient in doing that. And interestingly, we don't see this site in any other of these SARS-like coronaviruses. So this is a new, unique thing. So we looked a little closer at these particular features, this polybasic furin cleavage site, as well as this receptor binding domain. Now, the receptor binding domain, as I mentioned, it appears to be optimized for binding to the human receptor. But because people started sequencing more of these viruses from other animals, what we can see is that there's a couple of specific residues in this RBD, which determines binding into the human receptor. These are the ones that are in purple up here. But what we can see is that pangolins, viruses from pangolins, actually have exactly the same sequence as SARS-CoV-2. So what this tells us is that this ability of the virus to bind really well to the human receptor is actually something which is already occurring in nature, and specifically in viruses we've seen in pangolins. So we know that the reason why it's binding this receptor so well is actually because of natural selection, evolution in an animal host, because we see exactly the same thing happening in these particular animal hosts. Now, the acquisition of the furin cleavage site, so as you can see, this is just a picture showing like the alignment with other SARS-like viruses, and you can see right there, SARS-CoV-2 has this insertion that we don't see in any of the other viruses. It is still unique. We haven't actually found this in others, although I should say that some of the newer data from bats, we're starting to see remnants of this particular site. So we actually think that this is something that could potentially happen in bats too, that the virus gets these kinds of furin cleavage sites, of polybasic sites. The reason why this site is interesting is that if we're looking at these furin cleavage sites, what do they actually do to viruses? Well, we know from previous experiments that people have put these furin cleavage site into other SARS-like coronaviruses, not into SARS-CoV-2, but other similar viruses. And we actually know that that allows them to expand their host range. So now they can not just infect bats, but they can also specifically infect human cells. So we know that the furin cleavage site allows the virus to infect a new set of cells. We also know from the avian influenza field, for example, these viruses very typically pick up these furin cleavage sites too, and then make them more pathogenic. So that actually makes them more severe viruses. So if we go back to this picture that I had of SARS-CoV-2, I mentioned it could infect the upper airways, therefore spread really easily, the lower airways and the lungs to cause severe disease. We actually think that some of this has to do with the fact that it has this furin cleavage site, because it means that it can now bind to a lot of different cells. This ACE2 receptor is present in many different tissues, but they can also use the host enzyme furin to actually activate the virus and productively infect the cells. This is not something SARS can do, so SARS really stays low down in the lungs, and it's not something like the flu viruses can do either. So we think that probably some of these features that the virus picked up causes it to be able to be such an easy spreader, as well as causing severe disease. But that was just one part of it, because when we can look at these particular features, we can see them occur in nature. Obviously, that leads to some questions about, well, but how does this all get started? We know that it started from nature. We know that the virus is the effect of, is a result of natural evolution. But we have two specific hypotheses that could explain how all of this got started. Here's one that we call the market hypothesis. So this is linked to the Wuhan wet market, the Huanan market in Wuhan, where the bat infected some sort of intermediate host, where probably the virus picked up this furin cleavage site and then infected people at the market. And because of the acquisition of this furin cleavage site, now enabled it to cause human-to-human transmission really effectively. So we think that maybe this acquisition, I should say that it could also have happened somewhere over in the bats here, but somewhere along this way, the virus picked up this furin cleavage site and allowed it to become a pandemic pathogen. The other hypothesis that we also think is consistent with all the data is a pre-circulation hypothesis. So in this particular hypothesis, there's a reservoir, potentially an intermediate host, and then it jumps into humans, but it doesn't yet have this furin cleavage site. probably means that it doesn't cause very severe disease, so never have been detected. We wouldn't actually know that a new virus was infecting humans because they didn't get sick from it. And it didn't have this furin cleavage site yet, so it also didn't spread very easily between humans. But somewhere along that line, possibly in the human population, part of this undetected spread, it picked up this furin cleavage site along the way and then became the human pathogen that we then finally see now. So the timing on these things is that this could have been going on for a while, maybe a few months or so, and we just wouldn't actually have detected it. So these are the two hypotheses that are fully consistent with the data that we see. I know there's been a lot of talk about Chinese bioweapons and bioengineering and engineering in general. All of that, I can say, is fully inconsistent with the data. So we know that those hypotheses do not explain the data and can't possibly have been the way that SARS-CoV-2 actually arrived. So that's what we learned about how did this all get started. But let me just talk a little bit about where we actually are now, and also importantly, where I think we're going to go next. So a lot have been said about this, and I want to amplify this as well, because there is no question that the United States was sleepwalking into a pandemic. The United States, incapable of getting testing capacity set up, let alone just develop a very simple PCR-based test, really meant that for the longest, while we have at least 2 months to prepare for this pandemic to arrive on our shores, we failed to do so. Now, a lot has been said about this. I'm not going to go into the details about this, but I would say that this is a huge issue, right? We failed to look for the virus. We failed to actually prepare for the arrival of the virus, even though we well knew that that was going to happen. I think the main question is that do we want to continue to fail, or do we actually want to do something about it now? Now, one important implications about this, and I wanted to just show a tweet from one of my colleagues, Trevor Bedford here, because the Seattle flu study were actually the ones that detected, you know what, we have had SARS-CoV-2 in this country for a while. And the way that they did that was via sequencing. They had sequenced the case all the way back in January, and then they picked up a case later in February, and what they realized that one actually caused the infection in the other. So it's all connected. So what the data very, very clearly showed back then was that there had been transmission ongoing in the United States for at least 6 weeks by then, totally undetected, specifically undetected because we wasn't actually looking for it. So, and we all know this to be true now, but back then there was a lot of questions about whether that really could be true. But of course we know that this was absolutely true. Now, when we're talking about an outbreak in this country, an epidemic in this country, one of the things that's really important to keep in mind is that it's not just one epidemic. We have multiple epidemics going on. This is not like Italy or France, for example, where they're primarily dealing with a single epidemic. We are dealing with multiple, and multiple in different states. And this is just showing you some of that data from New York, New Jersey, California, and Washington. All got started about the same time, but as you can see the number of cumulative cases is very, very different between these different states. And it means that we have to deal with these as local outbreak, but of course as one joint effort as a country. Now, when we're looking at these outbreaks themselves, just going back to the genomes themselves a little bit, what we can show here is that the genomes, this is a tree, think of this as a family tree. The ones that are marked in red here are genomes of viruses sampled here in the United States. And what we can see is that actually there's only a limited number of sparks that led to the current US epidemic. So it's not like tons of virus got into the country and started the epidemic. It's a number of times the virus did come in. It was a limited number of times, and then the epidemic itself got going in the United States. And this is going to be important in a little bit later, so keep this in mind. Now, I already said this, fail once, do we want to fail twice? This is what we really need to plan for, right? Because we know that a second wave is going to come, a third wave, a fourth wave. We know that this is going to happen. The question we want to ask ourselves is that do we want this curve to continue to go up like this, or do we want to start seeing leveling off a number of cases like we have seen in many countries in Asia, and also starting to see in a couple of countries in Europe? So let me just look a little bit more into the future here and say, okay, how do we actually think that we can get this under control? And importantly, what are the timeline here? Now, I have hammered on about this. I want to make this clear. This is not the way to do it. right? This is the front page from yesterday. The irony here is striking with, you know, the deaths peaking in New York, a map showing all the many cases we have in the United States, which is by far the most in the world, while at the same time, we as a country chose to halt the funding to the WHO. This is not the way to fight pandemics, right? We really need to do this together. So I think it's exceptionally important that right now we come up with strategies, how we all jointly going to fight this together. But I wanted to address one question first, because I get this all the time. When does this end? This is a good question. The problem is, it doesn't. We have to really realize that we all want this to end, but in the short term, it's not going to. And the sooner we realize that, and the sooner we plan for that inevitability, the better it is. So what we need here are long-term plans. So if you look at some of these long-term plans, now the first part of this plan was to shut down. That was easy. That's why we're all sitting from home. That's why I'm giving this seminar from my living room. Shutting down is easy. Shut down, but the problem is shutting down only buys us time. It doesn't change the virus is still the same. Now, the talk in the United States, many other countries in Europe too right now, is when do we open up? That's also easy. Opening up is easy. You say, okay, let's just open up and see what happens. The problem is that what caused us to shut down in the first place hasn't changed. The virus is still there. The virus is still the same. If we open up now, we're just going to see an uptick in cases, right? And it's really important that we understand this. The only reason why we're shut down right now is to come up with strategies in which we can prevent future shutdowns. How can we actually live with this virus? So I'd like to think about this, not about shutting down, not about opening up, but finding a strategy for how can we remain open. We don't want to have these continuous shutdowns, right? Because it's very detrimental to the economy and everything else. So how do we actually get strategies to remain open? I just wanted to show this particular thing, which shows you why we are seeing cases starting to go down in many countries right now. It's because we're all locked down, right? Social distancing is actually working. If we don't get into contact with each other, the virus is less capable of actually spreading. So that's why we're starting to see cases going down in certain cases. It's not because the virus is different. It's just because that we actually have locked down, right? That is the effect that we are seeing now. And I like to compare this to, if you're thinking about building a levee with sandbags, if you're doing that and you start removing the sandbags, the water is still going to be there. So if you think about removing the sandbags, it's like us trying to open up again. The problem is that the virus is still there. Nothing is different around the virus, right? So you start removing these sandbags, well, guess what? The flood is going to come again. So this is the kind of scenario that we really need to prevent. And I just wanted to get back to this particular picture, which shows you that the limited number of sparks resulted in the current US epidemic. This is true in all other countries too, that the reason that other countries saw epidemics and outbreaks is because they had a limited number of the virus arriving on their shores and then causing a local epidemic. This, again, if we just open up our economy, open up getting back to work, it's not going to go away, right? The virus is still there. So we need to deal with that particular scenario. And I should say that it can actually be done. There are countries that have managed, especially in Asia, China and South Korea, I think are the best examples of this right now, starting to see it in Europe too, with countries that have been very aggressive in testing, very aggressive in tracing, very aggressive with isolating cases that are infected, as well as following up and quarantining their contacts. And I'm going to talk a little bit about this. But this is the way to get the curve down and hopefully have the curve stay down. There's no other way than us specifically fighting the virus and specifically suppressing the virus as much as we can, until we have herd immunity. The problem with herd immunity is that it can really only be obtained by a vaccine. Herd immunity is this idea that once, for this particular virus, about 65 to 70% of a population have immunity to the virus can no longer spread. The problem is that we look at the total number of cases currently infected and potentially immune. It's in the order of maybe 1, 2, or 3% or so. It's very, very low. There's no way that we can actually get to herd immunity by trying to get infected with the virus itself that we are trying to protect ourselves against. So we need to do that via vaccine. Problem is, vaccine takes time. So what do we have in the meantime? Well, here are the 3 primary knobs that we can actually twist and turn. One is social distancing. That's what we're doing now. That still is a way to combat this virus. Travel restriction, which we're also doing now, is another way. The problem is that both of these 2 are exceptionally disruptive, right? Especially things like travel distancing, social distancing, very, very difficult for just as a functional society. Social distancing is definitely going to be a thing in the future too. Things like, you know, can't do large congregations, can't do concerts, can't do rallies, can't do sports games where we all stack closely together. Probably just can't happen for the next foreseeable future. But the last knob here is to test and isolate. The idea behind this is that we need to test for the virus. We need to know where the virus is. And as soon as we get a case, we do something about it. We trace down the contacts, we isolate the people that are sick, and then we actually quarantine and follow up on the ones that might have gone into contact with that case. That is really the main knob that we have here and that we can use. So when we're looking at this, what do you actually think is going to happen here? And I should say that I am, you know, thoroughly unqualified on many of these, but this is going to take time, right? Right now, what we need to do is we need to test, we need to trace, we need to do isolation, right? It's our only hope right now. The problem is that we're not currently doing this very well. Most countries are not doing this very well. We certainly aren't doing this very well. So we need to do that. We need to hire contact tracers. We need to train contact tracers. Within the next year, I suspect that we will see things like repurposed drugs. These are drugs that are used for other indications and can actually be used against SARS-CoV-2. Drugs that are already in the pipeline, like remdesivir, for example, can be used here. Year 3, I think we're going to start seeing novel drugs coming up, as well as some of the experimental vaccines actually being rolled out. But I would say between year 3 and 5 is what we are hopefully going to get a vaccine. Now, I know a lot of timelines are a lot shorter than this, to say within a year we can have a vaccine. I think realistically, it's just not going to be possible. These things take time. We don't have a coronavirus vaccine. Vaccine designs typically takes about a decade. If we can actually have that, that is going to be pretty well. Now, Jamie mentioned this in the beginning, and I want to mention it too, is that we can think of these as 3 different pillars of things that we need to do. One is detect, one is cure, and one is prevent. And I just wanted to link it back again to the Scripps research and what we're actually doing here. There's the detect study, for example, led by our TOEFL that Jamie mentioned, have the outbreak.info and everything that we do around that, the cures for the drugs, the repurposing drugs, the pipeline drugs, and the prevent on, for example, the vaccine design. Now, Jamie mentioned this as well, but there's going to be a lot of upcoming front row talks that are going to talk exactly about these things, so you really should tune in to those. So with that, I just wanted to say, like, there's a couple of critical questions which I think we need to answer. Some of them are related to the type of work that we do. Many of them are not related to our work. Things like, what is the true exposure? We know how many cases have been confirmed, but we don't actually have a good understanding of exactly how many people have been infected. We can do that via serosurveys. I should say that we would expect the numbers here to be low. Low single digits is my best guess for what we're going to see here, but that is going to be important to know that. How does immunity work to this virus? How long does it actually last? Can people that have been infected once, can they actually be infected again? Are they immune? We want to wait for the clinical trials with things like remdesivir that I mentioned, as well as the IL-6 blockers. There's many, many other clinical trials going on, but these are some of the clinical trials we're seeing, just going to see probably within the next month or so. And I think at least the earlier things that we have seen for them are very, very promising. To say waiting for these clinical trial, which is only going to take about a month or so, is going to tell us, do we actually think we're going to have some drugs that we could use pretty quickly for this particular virus and are going to make a huge, huge difference. We want to understand things like some of these early vaccines. For example, Moderna has made a vaccine, which is now in stage 1 clinical trials. do people actually develop antibodies against the virus if they use that vaccine? What is the effects of seasonality? Should we expect the virus to go down when it gets hotter, for example? I should say that my expectation, we're not really going to see that. Effects of school closures is obviously very, very important to understand. Are these effective? Now, these experiments are already being done. Countries like Denmark, for example, opened up for the young kids in school earlier this week. We're going to see this as other countries are starting to do this too. What is the effect and the increase in the number of cases that we see? And more importantly, what are the rebounds that we see after relaxing these types of measures? So with that, I just wanted to finish by thanking, the type of work that we do here is an insane amount of collaborators. Here's just a very small subset of those. Very importantly, my own lab here at Scripps Research, and actually in other places of the world too, working exceptionally hard on SARS-CoV-2 and as well as many other viruses. I really cannot thank them enough. And then finally, I just wanted to finish with this initiative that Jamie mentioned, because the important when we're looking at, outsmarting emerging pathogens in an interconnected world, we're going back to these three different pillars: detecting them, preventing them from actually causing future outbreak, as well as curing people that get infected. And Scripps Research under this Global Health Initiative really covers all these three different pillars very, very well. And I'm pretty excited to see some of the research that we're going to come out of this. So with that, I am going to stop my presentation here. I think, hang on, no, I'm not. I'm going to pull up the Q&A. I should say that there is a lot of Q&As here. If you have a question, type it in. If I don't answer it, maybe write me an email, because currently we have 30 of them, so I don't think that we can get through all of them. But please feel free to actually reach out. Okay, so and I have to read these as we go. Okay, so the first question from Malcolm Ritter here, which is that some people have said the CDC stuck too long with the idea of requiring travel from China as a requirement for being considered a suspect case. So this question is really like in the very beginning of this, with very limited testing capacity in this country, the ability for doctors to test patients required them to actually have recent travel to China. If they didn't come from China, then they couldn't get tested. The problem was that by that time, there was already cases occurring inside the country here. So yes, this definitely delayed things, right? We had a lot of cases that were locally acquired, cases that obtained the infection here in this country, and we couldn't get them tested because they didn't actually have travel to China. So there's no question that things like that actually delayed our response to the virus. Okay, other than, this is from Paul Fisher, other than the S protein, what are other approaches cite might be effective inhibiting COVID-19? So if you're looking at this virus in general, the spike protein, which is the virus, that's the part of the virus that's on the outside, this is where antibodies will bind, neutralizing antibodies. So this is primarily what the vaccines will target, right? It's the S protein. What is interesting about this virus is it's very complex. It has a lot of genes. And every time a virus has a gene, that's a potential way that we could actually drug the virus. We can design a drug against that particular one, and then we can actually try and combat the virus. This furin cleavage site, for example, which we haven't seen in these viruses before, we have furin inhibitors, for example. We could potentially use some of those to actually try and combat this virus here. Okay. Okay, so I have Jamie Jensen here. I believe you said the reproductive rate for the flu is 1.5. You mentioned we must get COVID under 1. Why, if the flu is 1.5 and it is not considered a pandemic every year? Yeah, so this is a good question. So why is that? Now, it's because we have a vaccine. For flu, we have seasonal flu vaccines. And what that means is that we can protect ourselves against the virus, which means we can get that rate down below 1, so we can stop transmission that way. Because we don't have a vaccine for SARS-CoV-2, we can't actually do that. So that's the main reason why these things end up being different. Okay. Okay, so Rod Hale here is asking me a question. What evidence is there that COVID-19 were present in the USA in December 2019? Now, I know there's been some speculation on this via sero surveys here in California, for example. I should say that the evidence for this virus being in this country late December last year is zero. There is no evidence that actually shows that this virus was here earlier than January of this year. The reason why we know this is that a lot of work has been done on flu suspected patients going all the way back into last year. And when you do that, none of them actually show up positive for this particular virus. So we don't think that this virus was in December of 2019, but we do know that it actually ended up showing up in January of 2020. Okay. Okay, I have an anonymous attendee here which asked me how similar is pangolin's ACE2 compared to human. So this is pretty interesting, right? Because I showed you that this receptor binding domain that binds to the ACE2 receptor of the virus is exactly the same in coronavirus from pangolins as it is in SARS-CoV-2. So how similar? So you might expect that the ACE2 in pangolins might actually be very similar to humans. So I thought the same thing, and I actually looked at that particular question, and it turns out that they are definitely similar in some of the regions of the protein where they bind together, but overall not actually that similar, which I think is interesting. Now, there are people looking at this exact question at a much, much bigger scale. So I expect those studies to be out within the next couple of months or so. Okay. Gail Donahue is asking me here, how did the tiger in the Bronx Zoo get the COVID-19 virus? This is a good question because we know cats are probably susceptible to this. It seems like they're not really getting that sick from it, but probably the tigers in the Bronx Zoo got it from some of their caretakers that were infected with the virus. There's obviously a lot of human infections in New York, especially in Bronx at the moment. So likely that was one of the caretakers actually got infected and then managed to infect the tigers themselves. Okay, I should say that we're up to about 80 questions here. So I'm just reading through. Okay, so Che Emmanuel here is asking, for what you explain here, it seems there may be an increase in strains of SARS-CoV-2, which may make our efforts for vaccines meaningless. How do we see this? Now, I would say that now I know there's a lot about talking about different strains of this virus, but that's really not true. Viruses do diversify a little bit, right? They mutate over time, so we see some diversity. But importantly, it doesn't lead to new strains. So we are very convinced by now that one vaccine that would actually target one particular strain of this will target everything that we have actually seen during this particular outbreak. So we don't have any concerns in terms of that causing issues. Now, I'm just going to take 3 more questions, and then we unfortunately need to move on here because we're almost up to 100 now. Okay. Fane Robinson here is asking, why does it take so long to get a vaccine to market and scale? Now, some of the future talks will talk about this from Mike Farsan up next and Dennis Burton, I'm sure. One thing to keep in mind with vaccines is that we're talking about a vaccine that we would like to vaccinate the entire world. That's a lot of people to vaccinate. If you do that, you need to make sure that it's really effective and it's really safe. The unfortunate part of that is that it takes time. It just takes time to actually do that. We need to vaccinate people, see if they develop immunity, and are they actually protected against infection. So some of the main reasons why the first part of it actually takes time. And then of course you need to scale up manufacturing and things like that. Some of the main reasons that this is unfortunately going to take a long time. Okay, so I'm getting a lot of questions about like, could this has come from a lab? So let me just take that as my last answer here. We'll answer all of those at the same time. Now, we have a paper describing this. If you look at my Twitter, for example, we have described the paper, some of it in layman's term, and there's also a very short paper in the journal Nature Medicine. All of it is available on our website, so you can go check it out there. But the main thing that we do, the reason why we can show this is not from the lab is, number one, if somebody had engineered this, you wouldn't start with a bat virus, right? If you want to make a weapon out of this, or you want something that you're hoping to infect a lot of people with, you would start with something you know is already causing disease, specifically SARS, MERS, or whatever else you could think of, right? That would be your starting material. Clearly, that is not what this is. This is a bat virus, right? The second thing is that we're looking at these features of the virus, the furin site and this receptor binding domain, both of which we think is what causes this to be such a severe pathogen. Both of these particular sites we already see in nature. We've seen them in pangolins when we're talking about the receptor binding site. We've seen them in the bats when we're talking about the furin site. So we know that all these particular sites actually occur in nature, and very specifically exactly the same sort of sites that we see in this virus. So we know that this can all happen in nature, and we have seen it previously. The last thing is that, well, maybe somebody accidentally got infected in a lab, or maybe they've been growing it in tissue culture or something like that. We don't actually think that is the case, specifically because this furin cleavage site gets an addition of what's called old-link glycans, which is something that only happens when there's an immune system around. And of course, if you have a cell culture virus that you're growing, there's no immune system around. It's just the cell culture itself. And specifically, newer data also shows that these viruses, if you do grow them in culture, they actually lose this furin cleavage site. So we know that all the different scenarios that would be consistent with this having come from a lab, all the data is inconsistent with that scenario. It can't explain the data that we're seeing. But on the other hand, we can actually see that all the data is totally explained by evolution and their biology. So that's why we know that this came about as a natural virus that happened because of evolution in some animal hosts and not actually from a lab. So with that, I'm just going to finish here. I will give it back over to Jamie if you want to say a couple of final words.
Just want to thank you, Kristian, for a terrific lecture. And thanks to everyone who tuned in for the webinar. I hope that you found it was informative, and I urge you all to check back frequently on what we're doing at Scripps. Come to our website and stay tuned for an exciting series of front row lectures as we tackle this global health initiative. Thank you, everybody.